you very much, Professor Reddy, for this very kind introduction. Thank you for inviting me to give the talk. It's a great series that you've initiated. What a wonderful resource for geoenvironmental engineering or geoenvironmental engineering students and researchers and so on. The fact that it's recorded and kept, I think, makes it particularly useful. What I would like to do, I've got about 50 slides, and what I'd like to do is give you a sense of why questions related to microplastic and clay are worthwhile asking and investigating, and where does the complexity in these questions come from? So what does doing research in this area look like? That's really the aim of the talk. I will present some research results, but not a lot. So in that sense, it's not a conventional research seminar where I ask a question and I provide the answer at the end. It's more in line with what I felt was the spirit of the webinar series, is to give you an introduction, basically, into that area of research, focusing more on the complexities of the questions rather than research results that my group has generated. Acknowledging my collaborators, I'm sure many of you recognize most, if not all, of the names on this list, but my collaborators are acknowledged here. This is the research team which together we've been working on problems of basically microplastic and clay at the University of Sydney at the School of Civil Engineering for the past couple of years, perhaps 2 years and a half. My colleague, Dr. Elizabeth Carter, Dr. Suya Shudathan, and 3 PhD students, Yifei Gao, Liam Duncan, and Christine Fink. So the work that I'm presenting here is very much a collective effort by the team. This is roughly the outline that I'll be following. Just some background, what are the complexity, research, knowledge gaps and complexity, but I also want to zoom out a little bit at the end and look at the bigger picture on plastic. You know, as geoenvironmental engineers dealing with an emerging contaminant, the question of the bigger picture is always important. So what are microplastics? You look at definitions, quite easy in the literature. There's any plastic fragments that are less than 5 millimeter in size. When it comes to nanoplastics, some papers define them as anything that is less than 1,000 nanometers, while others define them as less than 100 nanometers. My sense is that most papers use the 1,000 nanometer threshold. There's an argument for using the 1,000 nanometer threshold in that below that size, colloidal behavior of those particles becomes quite pronounced, and that's pretty much the definition we've gone for, I and other researchers. There's a couple of papers that have actually were written to make the argument about the 1,000 nanometer, more or less. So we know, I'm sure you've heard on the news, if you've been reading any news over the last few years, that they've been detected everywhere in a range of ecosystems, including organs of the human body, but also in a wide range of ecosystems. And of course, the background problem is that after World War II, the production of plastic around the world has been rising pretty much exponentially. And correspondingly, of course, the waste produced as a result of that production of plastic is also increasing exponentially. And so microplastic, of course, comes from the degradation often of that plastic waste in the environment. Now, are they harmful? So you look at the the literature is now, the literature has built up, and there's strong evidence now of different kinds of harm. Again, ecosystem harm to ecosystem functions, to whether you're talking about oceans, about the biota in the oceans, whether you're talking about soil, and so on. But there's also, quite recently, we're starting to see evidence of link to disease associated with humans. This is one example, a recent paper in Nature, which has found a link between presence of plastic particles in blood vessels with possible heart attacks, stroke, or death. So we started to see that kind of evidence. This is very recent. So we started to see that kind of evidence. Now, you may be forgiven if you've been catching the news here and there about microplastic, for sort of having formed the opinion that microplastic is really a problem for the oceans, but not perhaps for soil or groundwater. And you will see that, in fact, a lot of the times when microplastic come up on the news, they often come up in the context of the ocean. Certainly the environmental dimension to it is often associated with the oceans. You find the person who coined the term microplastic, Thompson, in a paper from 2004, excuse me, is a marine scientist, and there's certainly been initially the community driving research on microplastic has been marine scientists, and this is reflected if you do a search on Scopus on microplastic, you do a search on microplastic generally with nanoplastic, and then you combine it with keywords related to different ecosystems. What you will find, if you look, for example, at 2018, to pick one year quite early on, you'll find out of 685 papers published in 2018 that have microplastic in them, 423 are about the oceans. 155 are about freshwater. For soil, it's only about 49. However, if you fast forward, and the plot at the below, in the lower graph now, or what I'm showing is the percentage of the total that is related to oceans, soils, or air, what you will find, if you look at 2024, you'll find now almost as many papers on microplastic in soils have been published as they have on oceans. Not quite, but almost. So there's certainly this is changing now, and although it started off with a lot of papers on oceans, there's a lot now being published on soil and to some extent on groundwater. So that's the first point about why evidence that microplastic is also a problem for soil and groundwater. But of course, the more immediate answer as to why it is a problem is that there's now a body of evidence that shows that microplastic have been detected in groundwaters, particularly under urban, industrial, and landfill sites. This is a list of studies from around the world: Korea, China, Italy, USA, Mexico, excuse me, which shows different concentrations of microplastics found in groundwater. And there's different kinds of microplastic, but mostly polypropylene, polyethylene, polystyrene, and so on. Now, we know also that we have a good sense of that there's a lot of evidence for microplastic coming into agricultural soil, coming through biosolid application, through atmospheric deposition. There's enough evidence for us to know that all this is happening. So we know it's present in groundwater, it's present in agricultural soil. Is it harmful to soils? And again, the answer is yes, a body of evidence is emerging quite fast. Initially driven by, or still probably driven by, soil biologists and people interested in soil-plant interactions rather than geotechnical or geoenvironmental engineers, but seeing quite a lot of disruption to soil function, to soil-plant interactions, and so on in the presence of microplastic. But what you find also, part of that literature, is you find that there's been evidence of microplastic, depending on the kind of microplastic we're talking about, depending on the quantity of microplastic, this effects on physical properties of soil. The kind of physical properties geotechnical and geoenvironmental engineers are no doubt interested in. So evidence of this paper's effect on soil porosity, the structural, the structure of the pores, the aggregate stability, the bulk density, water holding capacity, but also on hydraulic conductivity. All of this is there, which means that, again, this microplastic may be a problem for geotechnical and geoenvironmental and geoenvironmental engineers, because we are interested in those in the effect on those properties. But of course, the biggest argument for why geotechnical and particularly geoenvironmental engineers should be interested in that is, of course, that most of the plastic that becomes waste is landfilled, as that graph from our World in Data shows. The vast proportion, only actually quite a small proportion that is recycled, a significant one that is incinerated, but it's mostly landfilled or mismanaged, which means a lot of it ends up in sites, which often come under the design and thinking and control of geoenvironmental engineers. So it's certainly a problem for us from this point of view. Not surprising, when people went and looked at whether microplastic is present in landfill leachate, they found that it is. That's hardly surprising. There's plastic in landfills, and therefore it's not surprising that the leachate, which is the liquid that basically dissolves a lot of the toxic materials in the waste, in the solid waste, in the landfill, carries quite a lot of microplastic. Now, we know that, I'm sure a lot of people in the audience, if not everyone in the audience, know that the way we protect groundwaters under landfill sites is by relying on base liners. Different kinds of liners, but the simplest and arguably most common liners are the single composite liners, whereby you've got a geomembrane, usually a high density polyethylene geomembrane that's got good chemical resistance to the leachate. It can last long in contact with the leachate, and underneath it, usually there's a layer of clay of some kind, either a geosynthetic clay liner or a compacted clay liner. And underneath that is some buffer layer, and that's what separates the actual liner from the aquifer. The geosynthetic clay liners, which are now very widely used around the world, they're essentially made of two geotextile, a carrier geotextile and a cover geotextile, that sandwich a layer of very high quality sodium bentonite. High quality in the sense that if you hydrate it well, if you allow it to hydrate well, it can develop very low hydraulic conductivity, which is what you want in a lining system. You want very low hydraulic conductivity to minimize the likelihood of the leachate traveling from where the waste is to the aquifer. So the question that becomes almost very obvious is, how safe are microplastic, are aquifers and beneath landfills from microplastic and nanoplastic? Another way of asking the question, of course, is to ask how effective are particularly geosynthetic clay liners and sodium bentonite, because sodium bentonite is the most important component of the geosynthetic clay liners, at preventing the transport of small NP and MP. And I say small here because we tend to think that larger microplastic pieces are very unlikely to go through clay because of the microstructure of the clay. And so we tend to focus in our research on particles that are less than 5 to 10 micrometers. That's the focus. And these two questions are the driving questions for our research. Now, this is, I hope with the first part I've convinced you that it's worthwhile thinking about the interaction between microplastic and clay, and show you what are the driving questions are guiding our research, if you like. Now, what I want to ask is, I want to say, what do we know so far from the literature? What are the knowledge gaps, and what are the complexities of the research on clay? Let me start with, if you go and look at the literature on microplastics and clay, there's about 3 to 4 kinds of studies that you come across. Excuse me. The first one is where people look at the interaction between clay, often clay colloids, and microplastic in solution. So they mix them together and watch what's happened. Do they form aggregates or not? If they form aggregates, do they settle in the solution or not? This is really about basic interaction between those two components. And what we found, there is evidence in the literature that microplastic and montmorillonite, one thing I haven't mentioned that I'm sure most of you know, is that the bentonite that I mentioned earlier is usually made of around 70% of montmorillonite. So when we say bentonite and montmorillonite are not the same thing, but bentonite is made of, it depends on where you find the bentonite, but often it's made of about 70% montmorillonite. So of course, the research on microplastic interaction with bentonite is quite strongly related to research on microplastic interaction with montmorillonite. And so what we find, this is one paper, for example, which showed that in the presence of divalent cations, you get quite a bit of microplastic and montmorillonite forming heteroaggregates and settling in the solution. Something that doesn't happen if the chemistry of the solution is made of, is mostly ionic rather than cationic. Anionic, sorry, rather than cationic. And so that's one example of this kind of studies. Another kind of studies, when you look at the literature on microplastic and clay, is to look at how microplastic and clay compare when it comes to absorption of heavy metals on both of them. And that's a study that's showing that I think it's lead in this case, in this particular study, we're talking about lead, has a higher affinity to montmorillonite, particularly montmorillonite that has been colonized by microbes, has very strong affinity to that. It attaches to it quite a lot compared to polystyrene microplastic. That's another kind of study that you see in the literature. A third kind of studies is the effect of microplastic and nanoplastic on the properties of clay. What happens if you mix clay with microplastic and nanoplastic? What sort of change in properties? And here this is showing an increase in saturated hydraulic conductivity of the microplastic, depending on the size. This is polyethylene in this case that they've mixed it with, and they found some changes, quite pronounced changes in hydraulic conductivity as a result. The fourth kind of studies is where they look at column transport studies, and usually in sand columns, but they add clay either to the solution that contains microplastic at the source, or to the soil itself. In this particular case, it's been added to the solution, sorry, to the soil itself, I believe. And basically, you see some effect of the monomer. I beg your pardon. I think in this case, the bentonite was mixed with the soil itself to look at what happens. Does that increase or decrease the speed with which microplastic can penetrate through the soils? Another kind of study is where the solution itself that contained the microplastic is mixed with clay. And again, you look at the effect of to what extent it increases or decreases the speed with which transport through the sand is accelerated or not. And I hope I may have confused the two. There's two studies here, one of which they've added the microplastic to the solution, and the other one they've added the microplastic to the soil. It'll be clear if you consult the paper. It'll be clear which is which. The idea here is to illustrate the kind of studies that people have done on looking at clay. None of these, of course, allow us to answer the question about how effective are geosynthetic clay liners and a bentonite at preventing small MP transport. So the point I'm making is what we know already in the literature doesn't yet provide the answer to our questions. And so hence the drive to try and answer those questions. Now, I want to, in order to give you a sense of the complexities involved in this kind of research, what I'm going to do is I'm going to look at one of the most common ways of investigating problems of contaminant transport. Basically, if you're trying to find out whether a soil layer, how well it insulates or prevents the contaminant from running through it into what comes underneath, you can do, we've got, the geoenvironmental engineering community has a long experience, and the soil science community generally, in running those soil columns where you have an influent, an effluent. You can measure the concentration in the effluent over time, and that'll tell you how good that particular material as insulating against contaminant. So what I'm going to do is, so one way of looking at this is say, well, all we have to do to answer the question that I put on the screen here is to put a layer of geosynthetic clay liners in such a soil column and run have a solution at the source that contains microplastic and allow it to run, apply some pressure difference so that the contaminant would run through it, and then measure the concentration in the effluent and see how well the GCL insulates. So it sounds pretty straightforward. What I'm going to do is now in the next section of my presentation is show you why things are more complex than this. Use the soil column study as a way of illustrating what are the complexities of this kind of research when applied to microplastic and clay. So let me first, it's important to understand how we have used the soil column methodology, both as geoenvironmental engineers, but also as soil scientists. It's not just geoenvironmental engineers who use soil columns. Anybody working on soil physics will at some point have come across this kind of studies. So essentially, as I was describing, you've got your porous media. In this particular case, we're showing the influent being pumped from the bottom rather than from the top. And what you do, you pump it. Typically, you pump a certain number of pore volumes. A certain number of pore volume with a certain concentration, followed by a certain number of pore volumes with clean water. And in the meantime, you keep measuring the concentration in your effluent, and you plot C over C0 over time, or as a function of time, or as a function of the pore volume, and you get something like this, which we tend to call breakthrough curve. Sometimes you can also, at the end of your experiment, take sections and measure the concentration of the contaminant that stayed in the soil. Could be in the form of sorption, could be in the form of attachment to the soil grains. And you get this extra information in the form of a retention, what we call a retention profile or contamination profile. And typically the way you use this information is after you produce the breakthrough curves and the contamination profile, if you do get the contamination profile, you can create a model of the soil column and you use a version of the dispersion advection equation, usually the colloidal reactive dispersion advection equation. I'll explain in a minute what's the difference between the dispersion advection equation and the colloidal version of that. You inverse that you backfit basically the breakthrough curve and possibly the contamination profile to the results of your computer model. And based on this, you generate the parameters that basically describe, or at least allow you to test some hypothesis about what may be happening in the soil. Are you getting a lot of attachment? Are you getting a lot of detachment? And so on. And so from those parameters, and by repeating those experiments under different conditions, you can do inferences, theories, predictions, and so on. Now I'm going to talk about why, and often this is what we do when we first start investigating a new contaminant or a new porous material. That's typically the intellectual process that we go through. It's a mix of experimental and numerical reasoning that allows us to develop theories and predictive models. So what I'm going to talk to you about now is why this is more complex in the case of microplastic than it is perhaps in the case of dissolved contaminant, heavy metals or other contaminants. The first source of complexity with microplastic is that microplastic is not a particularly well-defined chemically. So just by saying microplastic, you haven't said much yet. There are different types of microplastic: polypropylene, polyethylene, polystyrene. There are different sizes. A microplastic only tells you that it's less than 5 millimeters, but there's several orders of magnitude between the smallest nanoplastic and the biggest microplastic. The shape of the particle may matter. The age of the particle may matter, and of course, the concentration of the particle may matter. Whereas when you're talking, for example, when you're studying lead contamination, typically, because your contaminant is well-defined chemically, all you need is the concentration. We're talking about dissolved contaminant, whereas here we're talking about a suspended contaminants suspended in water, whereas if you're talking about something like lead, it's dissolved in the solution. So all you need is the concentration to characterize it fully in your column. In this case, it's more complex because it's not as well defined chemically, the contaminant that you're working with. Even measuring concentration can be a bit tricky in that a lot of measurement methods work with number concentration, whereas other methods work with mass concentration. Some give you a number of particles. If you go back, for example, one thing you may have noticed, I'll go back to my slide on the presence in groundwater. You look at how this has been presented is in number of MPs per liter, rather than in the mass of MPs per liter. So this is to do with the characterization method. I will talk about that in a minute, where people count the number of particles they have found. Of course, those particles could be of different shapes and different sizes. So converting between number and mass concentration may not always be straightforward. Of course, there are other methods that measure the mass concentration. So again, that's another source of complexity. In terms of how do you measure those concentrations, or how do you characterize the microplastic in your sample, there are several methods. The most powerful, probably, rather, the most cost-effective and fastest is probably spectrophotometry, which relies on ultraviolet absorbance and reflectance. And basically, you apply an ultraviolet beam through your sample, and depending on the absorbance graph, you can link the absorbance graph to the concentration of the microplastic in your solution. And so you could plot a calibration curve, which you can then apply when you're running your experiment. The nice thing about spectrophotometry is that it's fast. relatively fast to do. It's reasonably low expense. The problem, of course, is that it really relies on you knowing reasonably well what kind of macroplastic you're expecting. So if you take an environmental sample and try to use spectrophotometry, where an environmental sample where you might have lots of different sizes and types of microplastic that you're not sure what they are, you might not be that successful with spectrophotometry. Also, any other the presence of other particles in the solution other than microplastic could interfere with your measurements quite easily. So you have to be very careful about that when you are applying it. Certainly the most powerful way of looking at microplastic, and one that is used quite widely, is vibrational spectroscopy. You could use Raman or Fourier transform infrared spectroscopy, and it's quite powerful in that it can tell you quite a lot in terms of type, size, shape, and even concentration if you do spatial mapping. Excuse me. Excuse me for 1 sec. As you can see, I have flu symptoms. I have COVID actually, with mild symptoms, but it does mean there's this inconvenience of coughing every now and then. Now, spectroscopy, on the other hand, is a measurement of every sample is quite time consuming, and it requires very high level of expertise. They're also expensive equipment and requires quite a lot of training. So they're not easy either to acquire or to master. So that's but they're pretty powerful. There are, of course, other methods. Pyrolysis, gas chromatography is used quite widely, which gives you a concentration in mass of the microplastic, the total concentration in mass in microplastic. There are other ways of doing things, dynamic or static light scattering, and so on and so forth. I won't go through all that. The main point that I'm making here is that one of the things you need to do is, depending on what is the research you're doing on microplastic, you need to get on top of your characterization. That's the first thing you need to do. How are you going to characterize your samples? It's not always straightforward. There's also still quite a lot of question marks about to what extent we can characterize nanoplastic. This is very much a field of active research that I think is advancing quite fast, and people can do quite easily. One of the things, for example, you will find when we looked at the studies of microplastic, people who have gone to take leachate samples from landfills and look at microplastic in there, all of them were actually quite large microplastic. We found no study. Things might have changed. So this is a very fast-moving field, but we found no studies which measured the microplastic in the leachate with the capacity to detect microplastic less, say, than 10 micrometers. And so that's still a gap, because the capacity to characterize smaller microplastic is very much an active field of research. So that was the characterization. My colleague Elizabeth Carter, Surya, and Christine, PhD students, are working on characterization methods. So this is part of the project that we're working on. So that's the first source of complexity, is how do you characterize. The second source of complexity is related to the soil column, is how well it works with clay. One of the problems is, often if you look at the literature on there's a very large number of papers that have done soil column studies with sand soil. In fact, the vast majority, what you're looking here, the graph that you're looking at, is the number of studies that we found in the literature, counting them, and the number of data sets, I beg your pardon, generated. So some papers have more than one data set. It's not the number of papers. The number of data sets that have done soil columns with microplastic, the vast majority really have very little clay involved. There's a couple of papers that have between 16 and 20% clay, but that's about it. And one of the reasons is that working with rigid soil columns with clay is a bit tricky because typically your flow rate is much slower. There's possibly the problem of side leakage is not always easy to control. And of course, a typical rigid wall soil column does not allow you to control effective stress. And so this is another drawback. And one of the things we have explored, is whether we can use flexible wall permeameters that are quite widely used in the geotechnical and geoenvironmental field, whether we could use them to conduct soil column studies with microplastic. And that's in order to do that, of course, you have to establish that your system is not acting either as a source or as a sink for particles that may interfere with your measurements. So you want to make sure that you've got mass balance, that the permeameter is not generating particles that are causing contamination of your samples. That's one research project that we are working on. The third source of complexity is that when we looked at the literature on microplastic and soil columns, we found that most of the studies only measure the breakthrough curves. Some papers report contamination profiles, but we found only one paper that has used the contamination profile in doing the inverse modeling. This is what this means is that there's a question mark as to how well the models in the literature capture the retention profiles. You see an example here where it's not particularly well captured in the literature. And so the question is, how important is the retention profile and not just the breakthrough curve in coming up with inferences and theories and predictive models? This is really to do with the viability of the inversion and the backfitting, and what data you're using in the inversion or what data you are not using. This is another question that is again that we are looking at. The fourth source of complexity is with the theory itself that underpins. And I don't have a lot of time to go into that theory. I think I have only about 5 minutes left. I've gone over time already a little bit. Essentially, the way we build the theory that would help us to interpret the transport of microplastic and other colloids in the soil is we tend to combine interface science and XDLVO theory with colloidal filtration theory. When we to try and interpret what happens in terms of interaction between different colloids in the solution, in the presence of larger soil particles. There's several processes that could be taking place: attachment, detachment, side blocking, brightening, and so on. And when we sort of upscale them to the level of the representative volume element, we get a colloidal version of the reactive dispersion advection equation. And the expression looks a bit like this. Those of you who have worked with the diffusion advection equation would recognize certainly the non-red part of the equation, the black part of the equation. And the red ones, if you like, are additional terms that are to do with what's happening at the colloidal scale. And so typically there's no way of measuring the parameters of this equation independently. And so what we tend to do, as I described earlier, is we tend to do those soil column studies and then invert this equation by backfitting the breakthrough curve to get those parameters, coefficient of attachment, coefficient of detachment, and so on. And that allows us to draw inferences. The problem, of course, if you look at the literature, there are multiple versions of these equations, in other words, multiple predictive models, and there is doesn't seem to be an agreement as to which model works under which conditions. It seems to be people tend to pick and choose generally. One of the problems is with the inversion itself, of course, in that when you are, often people use gradient descent algorithm to do the inversions, in other words, to backfit the breakthrough curve to the numerical model, the measured breakthrough curve to the calculated breakthrough curve, in order to find the parameters. They tend to do gradient descent algorithms. The most famous one is the Levenberg-Marquardt algorithm. And usually, if you've got a small number of parameters to backfit, that's not a problem. Your objective function, the objective function you're trying to minimize in the inversion process, tends to be reasonably straightforward. You've got a single global minimum. There's not a big problem. You can probably get to it with the gradient descent algorithm. But once you start having a larger number of parameters, then you might start getting multimodal objective functions. So there's always a risk that you might end up landing on a local minimum rather than a global minimum. And in this case, probably gradient descent algorithms are not the right way to go. That's another, even though that's what often been used, that's what is used most commonly. The other problem with a lot of the models is that they don't give you a measure of the uncertainty of your estimate of the parameter, so that they tend to be deterministic in nature, when in fact there's always a certain level of uncertainty associated with it. So these are all limitations that are quite significant, and the limitation that we are investigating. What I'm going to show you is quickly some results. I'm going to have to skip some slides because it's already almost, my 50 minutes are almost up. I'm just going to show you very quickly some results. We've managed to achieve at least 90%, around between 80 and 90% mass recovery of the microplastic in a permeameter, in a flexible wall permeameter. So we believe flexible wall permeameters can work quite well. We found that a lot of the microplastic has been held actually by the filter paper. So in these permeameters, you tend to place a filter paper, you tend to place your sample between filter papers to prevent soil grains from escaping, and the filter papers are usually then covered by porous stone. And what we found is a lot of the microplastic was held up by the microplastic. This is where we found well, held up, sorry, by the filter paper. The microplastic was held up by the filter paper. This is where we found the single most important component, most important sink of the microplastics. So we have a good sense of how to make the permeameter, flexible wall permeameter, work well as a soil column. In terms of bentonite polystyrene interactions, we've been running quite a lot of experiments, and we've been seeing quite a lot of evidence of attachment of bentonite of polystyrene onto bentonite particles. This is in the slide below. This is what you see as a background is a geotextile, and the bentonite attached to the geotextile, and the PSMP, the polystyrene microplastic, seems to have attached itself to the bentonite. So we're seeing quite a lot of evidence of that, but we're also seeing evidence of some microplastic. In this case, what we're looking at is the geotextile cover of a GCL with microplastic attached to it. These are all experiments that we're running through the permeameters, column studies experiments. We're also seeing some microplastic in the effluent, also with interaction with the clay. The other results that I want to show you are to do with the models and the theories. We've developed a new, basically a new hybrid method that allows us to identify the global minimum much more accurately, but also come up with an uncertainty, an estimate of uncertainty. We're combining particle swarm optimization with alternative ensemble Kalman filter for inverting the colloidal version of the reactive diffusion advection equation, and we've got some really nice results there that will be within the process of publishing. So you'll be able to see there's been a conference paper on this that you could consult, but we're in the process of publishing that in a journal paper. We also looked at a vast the database of all the results in the literature of soil column studies, and we have a better sense of which models work better than others. We looked at 8 models, and we find some have better predictive capabilities than others. We also found that under some conditions, having the retention profiles no doubt improves our confidence in our results. Here we're looking at the error that you achieve in the backfitting, depending on whether you have no retention profile, 7 retention profile points or 15 retention profile points. And so these are all based on synthetic data. So we know what the different parameters we're inverting are. And so we can compare to the actual results. And we find we get much better accuracy of our parameters if we are using the retention profile in addition to the breakthrough curves. Other research challenges, there's plenty. As I said, characterization of nanoplastic. We still struggle to identify environmentally relevant concentrations of smaller MP and NP, often because it's very hard when you get an environmental sample to look at the smaller MP and NP. We don't quite, I believe, understand. There's a lot more to understand, at least, about how plastic becomes microplastic in the landfill waste. And the struggle, of course, the biggest struggle is the fact that we've got a vast we've got a vast diversity of plastic types and different shapes. And as plastic ages, it changes quite a lot in property. So if you're interested in microplastic, there's a lot to do, and there's going to be a lot to do, I'm sure, for the coming decades. To conclude, just a quick point about the bigger picture. One thing is to realize is that recycled microplastic accounts for only a very small proportion of the microplastic waste, what happened to microplastic after it is produced. And what you'll find is, the vast majority of the plastic is used once. So out of the 8,300 million primary plastic, tons of primary plastic produced from 1950 until 2015, 5,800 millions have been used only once, and therefore most of them have gone to landfills. So it's an incredible proportion. Recycling is very small. If you think that by recycling is solving our problem with plastic, you obviously you're far from the truth. There's a big debate about, there's currently negotiations happening about whether to have a plastic treaty, and some of the questions that are being asked, some of the disagreements that are there are about whether first to drastically reduce production and therefore consumption. There's an argument that we need to reduce production and consumption. If you look at the per capita waste generation, you will find that it's probably in places like Germany, USA, Ireland have high per capita plastic generated per day. But some people argue that what matters more than the production, that people argue that we're probably looking at the wrong figure there, and the figure we should be looking at is share of global plastic waste emitted to the ocean. That's one thing, because the ocean has been at the forefront of, ocean scientists have been at the forefront of fighting plastic. And here the picture shifts interestingly. If you look at this map and you look at the share of global waste emitted to the ocean, you'll find India, China, and Brazil become important players. So here, people who argue this argue that what we need is the development of effective waste management infrastructure rather than a cut for production. And of course, negotiations are happening. And as you can imagine, in a treaty of this kind, there's a lot of different interests. Different countries are still developing their position on this. And while there was agreement in March 2022 that the countries have agreed that we need a treaty, this is now, negotiations are happening now, and it's all culminating in Korea in November of this year, where we're supposed to sign a treaty. But reading the literature on where the negotiations are now, there seem to be quite a lot of obstacles still to overcome. And with this, I finish my presentation. My apologies for having gone over time.
No
I will stop
problem.
sharing.